for the last presentation, which is from me in this session, kind of connecting some of the dots that you've been hearing about up until this point, here focused on what we call the ERGA Community Genomes. So I think you will have seen in the presentations that came before that the genome generation workflow contains many, many different steps where you can go wrong, where you can fall down if you don't have a helping hand along the way. And really, this is the motivation of the work behind building the ERGA community, to be able to immediately find someone who has experienced these challenges before, who has maybe found a solution and can help you overcome your issues, all the way from the very beginning of the stages, through the sampling, through the extractions, through the assembly. You just heard about the reviewing process to ensure quality, all the way through to the data deposition in the public repositories. And really, you know, how this happens and where this happens, and you heard there briefly from Diego about the work of the Sequencing and Assembly Committee as one of these pillars, is the committees in ERGA, where these are the places, these are the forums where we are trying to develop these community standards that are agreed upon by participants so that they are realistic and achievable. But also practical things like best practices documents, practical guidelines. You also saw some examples of bioinformatics analysis workflows where Diego even said there that they've been ported onto Galaxy so that they are widely accessible and that everyone can be using the same kinds of workflows, the same kinds of standards, so that we're talking the same kind of language, so that we know what we're talking about when we are addressing things like assembly quality. So metadata you heard from Astrid's presentation earlier, information sharing about what works and what doesn't work. And this is really the fundamental work that is carried out by the ERGA committees. For example, here as a kind of starting point for the community genomes, we put together a list of questions that a researcher might ask themselves at each different step along the process. Again, with some answers to those questions, but usually directing to the relevant committee where that question might be posed and hopefully someone can answer. So for example, just under point number 2 here, under sample acquisition strategy, have you thought about your permits and ABS? Have you thought about your permissions and your material transfer agreements? How are you going to do your taxonomic validation? Do you know what is specimen vouchering and where you might be able to do that? Have you thought about where you might send your leftover samples for biobanking? Do you know the best conditions for shipping? So for example, after you've done your collections, to send it to the sequencing center. You can ask these questions, find the answers so that you don't end up shipping it incorrectly and your sample arrives at the sequencing center in a pool of melted ice. So that's just one example of the kinds of things that we're trying to implement to help community build their own reference genomes. Part of this is, of course, also coordinating efforts across different projects, across different research groups, in our case, at the European level. And so this view here that you can see on this screen is the portal, ERGA Biodiversity portal, which is tracking biosamples created at the European Nucleotide Archive, which Tyler mentioned earlier. So the projects that are coming up, the samples that are arriving, and the data that are arriving, and eventually the assemblies that are arriving, so that the entire community can see at what stage the generation of a reference genome is at for different species being produced by a whole group of different projects underneath the umbrella project of the European Reference Genome Atlas. And for example, another important reference resource, if you like, for tracking and coordinating efforts across Europe is the Genomes on a Tree resource that Tyler also mentioned earlier. And here just an example of specifically the Biodiversity Genomics Europe sub-project of ERGA, showing more or less the progress in terms of what has been collected, what is undergoing sequencing, and what is moving into the assembly process. And so how, is the best way to begin to engage with the community coordination efforts? And really this boils down to 2 main points, is how to communicate which species you or your research group is working on at the moment. And then at a second stage, how to get your genome ERGA approved, which is something that Diego was addressing there with this assembly review process. So first of all, communicating which species you're working on. This is mainly through the Genomes on a Tree spreadsheets. And at the kind of ERGA coordination, we're trying to centralize this to help people to achieve this more easily. So either through the spreadsheets that are as part of the BGE project or the pilot project, so ERGA core projects, or other European projects that some of them were mentioned today, the Catalan Biogenome project and others that are related and connected to ERGA. And what's currently under development now is building country-specific sheets for Genomes on a Tree, so that each country can really showcase to the world the different species they are working on, or the research groups that which taxonomic groups they're focused on. And in parallel to that, building what we call the ERGA Community Genomes on a Tree sheet, while we're still getting established to have individual country GOAT sheets. In terms of the second stage, which is maybe you've already produced your reference genome and now you want to get it ERGA approved, here it's really two simple validations that we are implementing at the moment, which is the first one, the metadata validation check. So this is basically the samples and sample processing committee checking that your metadata that you've collected meet the minimum required standards to be considered an ERGA approved genome. And the assembly validation. So those are the EBP standards that Diego was just presenting there. So the sequencing and assembly committee being in a position to check that your assembly meet the required standards. Then we can move or connect your bio project on the European Nucleotide Archive underneath the ERGA umbrella and giving it that ERGA seal of quality or ERGA approval, if you like. And finally, how to do this. So when you get stuck, when you have questions, really the main take-home message of this session is that there's absolutely no need to struggle all alone. We have within ERGA our Keybase community chat platform, and we also have for all of the different committees group mailing lists. So those are kind of your two main avenues of communication with experts who might be able to help you answer your questions to overcome your problems. So this is also to reiterate that you don't have to be a regular committee participant. So you don't have to be turning up at every single monthly committee meeting in order to ask for help. So through either the Keybase community chat platform or through joining the mailing groups of each of the different committees, you can post your question on Keybase in the committee's relevant channel, or you can email the committee steering group, which each committee has its own email address, which you can find clearly on the website. And indeed, you could even perhaps request a slot in an upcoming committee meeting where you might present some of your data and say, this is where I've got. I don't understand what these graphs are telling me. Could you help me understand if I need to go and do more sequencing or if there was something wrong with my sample? And there's a group of experts, and amongst this group, hopefully one or two of them may have seen something like this before and can give you advice on how best to proceed. So if you haven't signed up to join ERGA already, then hopefully you are motivated to do so. And the channels there are really easy in the Keybase community chat platform and the committee group mailing lists if you want to become more engaged. And you can also just browse on the website for the time being to find out the kinds of events, the calendar of events indeed, for when each of these committee meetings are happening. So Chiara started off by saying we welcome interaction, we welcome participation, and I guess I can kind of close the circle now with the reiteration of that message. And thank you all very much for joining us this afternoon.